Good afternoon to all of you. Today we are going to deal with the last heading of the chapter, reproductive health, and you are going to learn a very new topic which you have not learned in your previous classes. So we are going to learn about infertility first. So inability to conceive or produce the children even after 2 years of unprotected sexual contact is basically referred as infertility. There are several reasons for infertility, like physical and congenital diseases, immunological problems. Then the male infertility, the main reasons for the male infertility are reduced sperm count, absence or extremely low count of motile sperms, absence of penis, failure of testes to descend in the scrotal sac. These are the reasons for male sterility. Same way, there are reasons for female infertility also, that is non-functional or absence of ovaries, non-functional or absence of uterus. Fallopian tubes are either damaged or ligated. So this is basically the reason for infertility in males and females. To cure this infertility problem, there are certain techniques available, and we are going to learn these techniques under the heading that is ART, assisted reproductive technology. In India, often females are blamed when the child is not produced or born. So there are many infertility clinics that are helping in treating this disorder and enable a couple to have their children. So let us look out for the techniques. So it basically the art include the medical procedures that are dealing with infertility, IVF, then ICSI, that is intracytoplasmic sperm injections. There are few. Now let us see what are the techniques or the types of art. IVF, that is in vitro fertilization, IUI, intrauterine insemination, intrafallopian transfer, then ICSI, that is intracytoplasmic sperm injection, then gamete, intrafallopian transfer, that is gift. So these are basically the different types of assisted reproductive technology. We are going to deal with these technologies one by one. Now the first is the IVF, in vitro fertilization, which is also known as test tube baby. Here the fertilization is done outside the female body in almost the similar condition as it is present in the within the body. Now here the embryo is transferred. The ovum is taken from the wife or the donor, and sperm is taken from the male or the donor, and in a lab in sterile condition, the male and the female gamete are fused to form the zygote. This zygote then start changing into early embryo, that is up to 8 blastome years, and it could be transferred into fallopian tubes. When it is transferred in the fallopian tube, it is known as zygote intrafallopian transfer, that is ZIFT, ZIFT. Embryo which has more than 8 blastome years are transferred into uterus, and that is known as IUI, that is intrauterine transfer. Next technique that you are going to learn is the gamete intrafallopian transfer, which is GIFT, GIFT. Now as per the name, here the technique is used for the female with the prolonged sterility, and they should have at least one potential fallopian tube. Ovum is collected from the donor female and is transferred into the fallopian tube of sterile female, which is a recipient that cannot produce the ovum. So here the potent female is gifting the gamete, that is why the name is GIFT, GIFT. Next technique that we are going to learn is the artificial insemination. It is usually used when male partner fail to inseminate or suffer from reduced sperm count, that is oligospermia. Semen from male partner or donor is collected and is introduced into the vagina of female, and semen is introduced into uterus also in certain cases. So it is intrauterine insemination. and when it is introduced in the vagina, then it is known as intravaginal insemination. The last technique that we are going to learn here is the intracytoplasmic sperm injection, that is ICSI, the last technique which is shown here. It is another type of specialized process where embryo is formed in lab, the sperm is collected and directly injected into the ovum cytoplasm. That is why the name is intracytoplasmic sperm injection. The zygote further can be transferred into female reproductive tract for further development. So these are the techniques of art that are used by the infertile couple in order to produce their own babies. There are many drawbacks of these techniques. Let us learn for the few of them. The major drawback is that they require very high precision and they are very expensive. The next drawback is there is no surety for the 100% success for these techniques. Now, the last drawback associated with the art is that if the uterine wall in female is damaged, then none of the technique can be practiced. So, this is all about the reproductive health and the art that we have learned today. Kindly go through the video properly, and in case of any doubt or a query, you can call. Thank you.